Well, good evening, everybody. Thanks for coming along this evening. I'm Alistair Burt, I'm the Executive Dean of the Faculty of Health and Medical Sciences. I'd like to start by acknowledging the Kaurna people, the original custodians of the Adelaide Plains and the land on which the university's premises at North Terrace, Waite, Theberton and Roseworthy are built. So it gives me great pleasure to welcome you to the first of the ED lectures this year. We've rebranded them as Health Matters. As I say, thank you for joining us. I'm delighted to introduce Professor Hamish Scott this evening. As many of you know, Hamish heads the Department of Genetics and Molecular Pathology at the Centre of Cancer Biology, which is an alliance between SA Pathology and the University of South Australia. He did his PhD in Adelaide and his first postdoc at Adelaide Women's and Children's Hospital, followed by 5 years at the University of Geneva Medical School. He's led many international collaborations to identify genes causing a number of diseases, from severe birth defects to familial cancers. He's been central to introducing a new genetic testing technologies to South Australia to personalise patient treatments. So to discuss Your DNA Matters from Conception to Old Age, delighted to introduce Hamish.
All right, thanks very much for that. If I sound a bit croaky, it's because I've had a bit of a cold. I did have matters in the title before you actually changed it to Health Matters, because your DNA does matter from conception to old age. And I have given it a more scientific title, if you like, which is about human genetics and genomics, DNA testing in both research and healthcare. And so I'll try and give you a flavour of where we're at in standard care and also in research at the moment. Of course, your DNA actually matters well before conception. If you're choosing a potential partner to breed with, it might matter. And it's not going to be unusual in the future, probably, that you'll have your genome readily accessible and it might be part of your dating profile. So everyone who laughed at that has a secret. And no one else does. Okay. So I just start with some acknowledgements. A lot of funding bodies to basically the whole body of work I'm going to present to you today. A lot of people, particularly associated with some of the work I'll present on fetal DNA and fetal autopsies, Alicia Burns, Sarah King-Smith, Per Arts and Chris Barnett as a lead clinician. A lot of work getting a lot of what we've done into routine diagnostics, particularly Karan Kassan and Janice Fletcher. And a whole bunch of people involved in the cancer work that we've been doing. Anna Brown, Chris Hahn over the years, Richard Deandra, Devendra Hawasi, there's too many people to mention. So I thought I'd actually start with a thought for you, and I may well finish this with this thought as well. In Australia, we've been lobbying hard since I can actually remember, and that's almost 30 years now, for an increase in research budgets, largely to no avail, except under the doubling of the NHMRC budget under Howard. So in the health arena, with approximately 1.6 of research expenditure, Australia generates 3% of world publications, and our citation rates, meaning the quality of our work, is actually higher than many other countries as well. And you can actually get this comment from politicians saying, well, if you're only generating 3% of the data, then why bother at all? Right, so 3% of it, 3% of patents, 3% of papers, why don't we just leave it to everyone else? Leave it to America, for example, or leave it to China. And my main response to that, because if you want to actually benefit from the research that we do socially and economically, as soon as possible or immediately, and from the research being done elsewhere, you actually have to be part of generating it. And I hope I'll show you some examples of that. So we're going to be talking about DNA and our genome, and there's some subtleties in what I've written in the text on here and what I wrote in the abstract. Almost every cell in the human body contains 2 copies of a human genome. Your gametes have only 1 copy and your red blood cells have none. So a human genome is actually roughly 3.2 billion data points. Okay, so in every cell you've got roughly 6.4 billion data points. It's got 4 different chemicals in it. And these chemicals called nucleotides make up a very long molecule called DNA. So each cell has 2 copies of 23 long DNA molecules, which are actually called chromosomes. And on those chromosomes, there's roughly 20,000 genes that encode proteins, which make the physical building blocks and machines of a human. So of those 20,000 genes, that's roughly only 2% of the genome, and we know what some of that 2% does, and a lot of the rest of it is actually still a black box. So there's a lot about our genome that we still don't understand, and there's a lot of it which is actually repetitive, and we really have no idea how that works and why it's there. So that actually seems like a relatively simple equation, and in fact we don't have that many more genes than a small nematode, a small worm with only 800 or so cells. So how are there so many different cell types and with different functions? What I've presented you is a very static and binary vision of what a genome is, with 6.4 billion data points in a cell. It's not like that at all. It's not linear, it's three-dimensional and complex. And to try and give you some idea of that, I recommend you go to this animation site for this guy called Drew Berry, who I used to work with at the Walter and Eliza Hall. He does some amazing animations, and some of them have actually received prizes, such as the British Academy Awards for filmmaking. So this is an animation of DNA. So here you see a DNA double helix. Many of you would have heard of a DNA double helix. So this is it in its linear form, and each little dot there you can see is a nucleotide. But you see it's moving, so it's not static. More than that, it actually wraps itself around proteins. These are called histones. And each of these proteins have little tails hanging out of them. And those little tails are actually, if you like, environmental sensors. And they can be reprogrammed depending on what you're doing, depending on what you're eating, what you're drinking, how much exercise you're having. They're modified by things like methyl groups, which are found in the folate you might take before you're pregnant, or folate in green vegetables. So yes, you should be eating your green vegetables. So you see the DNA isn't linear, it's folding up into these complex structures, and the way that it folds up can actually alter how it expresses itself. So how those 20,000 genes in the genome are expressed, how they're expressed as RNA, and how they're expressed as protein. So you see these structures are going to get more and more complex as the DNA is actually compacted into a form which many of us are familiar with, called chromosomes, which you've just seen there. And every cell has to divide. So this is actually photography of a cell dividing, and what you'll see is that the chromosomes have replicated and they're now lining up along the middle of a cell. Whoops, I've stopped it. That's very annoying. And in a minute, they're about to be pulled apart and the cell will split. So this is a DNA replication called a mitosis. I'm sorry, that seems to have stopped short. So, okay, there's a DNA replication. So the point is that we start off as a single-celled organism where a sperm and an egg are combined, and we have numerous cell divisions after that, which happen over time. So you can actually inherit a mutation, and so have a mutation in your DNA in one of those 6 billion data points that you got from your mum or your dad. Or you can actually get them during these cell replications as they go along, or they can be invoked by environmental mutations. So agents such as benzene, cigarette smoke, obviously radiation. As we age, we actually automatically accumulate mutations. So that's partially because of our environmental exposure, and that's partially because the system isn't completely perfect, although it's pretty bleeding amazing in terms of replicating things perfectly every time you have a cell division. And so as we get older, by the time we're about 80, there's probably 1 in 2 of us who are suffering from cancer simply because we've accumulated a whole bunch of mutations and it's too much for the cell and it starts hypermutating and hyperproliferating. Mutation. What does a mutation mean? A mutation can be as simple as one single data point from those 6.4 million data points which alters the function of one of these proteins. It can be more subtle than this. It doesn't have to alter the function of a protein, but for simplicity we're going to say that it alters the function of a protein. Maybe it cuts the shape of one of these building blocks or the size of one of these building blocks in half. And that can be enough to cause a severe defect at birth, something which manifests a little bit later in life, or indeed a cancer. It can be more complex and can include a lot more of the data points than those 6.4 million. So if you've heard of Down syndrome, for example, Down syndrome children have a trisomy of a whole chromosome, but it's actually, so they've got about 1% more of the whole human genome than most other people do. So how do we actually end up knowing all this? This is a photo of Gregor Mendel, a monk who performed lots of experiments with peas back in the 1800s. And so you might have heard of Mendelian inheritance. This is named after him. But really a lot of the knowledge that we've got at the moment and the power we've got in human genetics and genomics comes from knowledge aided by the Human Genome Project. So the Human Genome Project was remarkably controversial when it was proposed. And it cost roughly $3 billion over 15 years. And there's some comparative pricing for you. I haven't put the Joint Strike Fighter up because no one knows how much it's going to cost yet. The funding actually came from NIH, which is the National Institutes of Health in America, but it also came from the Department of Energy in America. So why would funding for a project like this come from the Department of Energy? The Department of Energy actually used to be called the Atomic Commission in America, and they were actually after techniques to measure how DNA was changed by radiation. Because they'd noticed, for example, after you drop a couple of atomic bombs, there's an increased risk of cancer, and so they wanted to understand that. So the genome was announced as finished by Tony Blair in the background and Bill Clinton in 2000. It turned out to be a race between the public and the private. This is Francis Collins, who headed the public effort. He's now head of NIH. He's survived Trump. And this is Craig Venter, who headed the private effort and wanted to make lots of money and patent everything. So this man here, he's been recurring during genomic science. He's very persistent. It's one way of putting it. He's stark raving mad, and incredibly ambitious is another. Guess whose genome he sequenced when he was in the private effort? It was largely his own genome. He was also responsible for sequencing the first domestic dog. Guess whose dog that was? A man of no small ego. So since that happened at a cost of $3 billion, they estimate that the first human genome, the draft of a human genome sequence, cost $100 million. And since then, particularly since the late 2000s, there's been an absolutely massive drop in how much it costs to sequence a human genome. In fact, this has gone a lot faster than Moore's law of computing, which is halving in price and doubling of processor time, which actually causes some problems with the massive data sets that we generate in genomics now in terms of being able to have sufficient processing power. Now, they keep on updating this slide, but I've stopped bothering because it's got down to roughly $1,000. But this is a bit of a lie. You might have heard of the $1,000 genome. What they're saying is that's the cost of consumables to put on a machine. They're not counting the cost of doing an analysis. And so how much does it cost to actually store your data, think about it and work out what the heck it means, because you've got 6 billion data points. But how did we get to do that? Well, there was a massive boost in miniaturization and parallelization. So part of the Human Genome Project was actually focused on technology development, and it worked very well. So we've gone from working in tubes like this to microcapillaries like this. So we've got strips of 8 to strips of 96, where we literally have what's called massively parallel sequencing, where we're just throwing DNA onto a microscope slide and running thousands of reactions or millions of reactions on one slide. So this is a history of some of the technology, and you can see how it's developed. This was the first groundbreaker back in 2005, which allowed a whole human genome to be sequenced for roughly $1 million. And there's been a slew of these different technologies afterwards. Now, if you remember, I said the $1,000 genome was a lie because you didn't count the cost of the analysis. It also doesn't count the cost of buying a machine and keeping your technology up to date. So there's no amortization if you're going to be a businessman about it. It didn't stop in 2012, 2014, 2015. The Garvan Institute invested heavily in this massive system called an Illumina HiSec X10, which was roughly $10 million. And yes, it could do a human genome for roughly $1,000 in consumable costs, and it had the capacity to do something like 18,000 human genomes per year. And there's several of these machines throughout the world, so we're probably in a position, if we really wanted to sequence massive populations, and that is occurring in some places. This is one of the latest machines that we bought. It's called a PacBio. You don't need to see all the detail. It's a machine which is about the size of a big freezer, $800,000. Its technology is actually better than some of the other machines, but its consumables for sequencing a whole genome cost $30,000. Right, so it's a better quality genome that you get out of it, but it's more expensive and the machine is still costly. One company has become the predominant provider of most DNA sequencing technologies across the world. We have one of these in Adelaide, we have a couple of these, we have one of these, we have one of these. What we desperately need is one of these. So these came out roughly 18 months ago. So if you've got a lazy $1.4 million hanging around, come and see me with your checkbook afterwards, because this is what we need to stay competitive in South Australia at the moment. There's only a couple of these in Australia. So are we going to be sending DNA into massive centres in the future? Well, who knows? I told you this all came about because of miniaturisation, and that miniaturisation continues. So this was the release of a couple of machines in 2014, 2015. This one was the size of a lunchbox, and as with many of these technologies, they're actually superseded before they get to commercialisation, and they don't make a huge impact on the market. And so the amount of venture capital being sunk into this technology is amazing. And this is the latest technology from one company called Oxford Nanopore. So in the future, you might be sequencing your genome at home. Okay, it's not beyond the realms of probability, and the small personal device that you have, whether it be an iPhone as depicted here, or some other device, might be actually doing your DNA data analysis for you. So who cares? We can sequence human genomes. Well, we started out pretty well. We did a few celebrity genomes because after we'd sequenced Craig Venter and his dog, we wanted to know what made Ozzy Osbourne tick. We did Desmond Tutu. We did a few other celebrities. But really, that's not my cup of tea. I prefer sequencing serious medical problems because this is where the power is in the immediate. Oh, you may have read recently that the twin astronauts that had their genome sequenced, the way some of this gets distorted in the press is also amazing. These twins were actually said in the media to have 7% of their genome different once the twin had spent a year in space and came back. This actually shows the correction that NASA issued saying, no, they're still twins, okay? The DNA is still identical. It was 7% of those 20,000 genes are expressed a bit differently, so it's quite a different story. But going into space, that does alter your environment, so it does alter the way your genome is expressed. So there are all of these large-scale genome projects going on with massive amounts of sequencing. So there's a massive amounts of sequencing going on in cancer to try and understand why cancer occurs and how you might be able to treat it. Genomics England started sequencing roughly 100,000 people to try and work out rare diseases and also try and follow populations longitudinally to develop increasing health data. Craig Venter, he's still around, he's still doing lots of things. He's got an institute named after him, of course, the Venter Institute. He's got a company as well, and they're sequencing really old people, trying to work out why people can live long and healthy. So there's lots of different things you can do with this technology. But in terms of politicians, David Cameron was one of the first people to recognise the power of this. And that's probably very likely because in 2003, he had a child who was affected with a rare genetic disorder called Ohtahara syndrome, and his child died at 6 years of age. So if you want to make a change in this type of field, just have a politician who's affected and has something to actually gain from these technologies, or then has an understanding of what might be gained by these technologies. So what were we doing here? We had some of these machines. Actually, before we had these machines, we started to think about what we could do. So we know that personalised genomics is going to be on the rise. It's relevant to trying to work out why people might be intellectually disabled, what inherited diseases they have, for pharmacogenomics, carrier screening, prenatal testing, familial cancer, and oncology. And of course, the best thing that you can do in medicine is actually work out how you should be treating someone. So if you've got a diagnostic test is positive and that you know you should be treating someone, that is the most effective and normally the most economical way of doing medicine. And so genomic testing has a massive possibility in this area. So this is the way that genetic testing used to be done largely for children. A child would present to a genetic clinic, and the clinician would look at the child and look at some of his laboratory features, and maybe if the child was slightly dysmorphic, having a slightly abnormal face, he would try and work out what gene he thought was responsible for the disease by their acumen, by their experience. And then they'd order a gene test for roughly $2,000, and it might come back negative. Come back positive sometimes, it might come back negative. And so then they'd go on and order gene 2 if it was negative, gene 3. This is what was called a diagnostic odyssey. And many patients went through, and their families went through these diagnostic odysseys. And you can imagine that at roughly $2,000 a gene, started out more expensive than that, but say roughly $2,000 a gene, the turnaround time to actually had a diagnosis and so you know what you're meant to be treating your patient for was actually massively long and extremely costly, and what those patients and their families should be doing is very difficult. So the ability to sequence all genes of the human genome, or a whole human genome at once, has basically transformed genetic diagnosis, so you can sequence all genes at once and then analyse it and see what's wrong with the child. Easy, you say. Well, it's like finding a needle in a haystack. So if you actually look at the person next to you, unless they're from a totally different ethnic group or from the greater apes, you'll probably have roughly 4 million different sequence variants between you and that person. Which one is the one which causes what you want to call the physical or phenotypic difference between you, or maybe some other phenotype? What causes that person to vote Republican? God knows. Or what causes them to be musical? These are the types of phenotypes. So actually working that out is very difficult. And there's clearly a level where we can go to in a diagnostic lab, and then when it turns into research, because we reach the end of our knowledge at that time. So we actually started using whole genome sequencing, and this was one of the first families in South Australia that we did. And the parents of this small child who has a disease called diamond blackfan anemia set up a foundation, and they raised over $4 million to research the causes. And they spent $20,000 of that accessing whole genome sequencing very early when whole genome sequencing was available, so it was still at $20,000. He was actually diagnosed at 8 weeks of age, and he has a transfusion-dependent disease. And so he was transfused over 100 times by the time he was 7 years old, I think. And he's also at increased risk of developing cancers, including MDS and AML. So this was sent to Illumina, I shouldn't have said that, the big American company, to try and work out what was wrong with him, and his sequence was sent back because they couldn't find out what was wrong. Their searching for the needle in the haystack didn't actually work out what was wrong. We actually did work out what was wrong with him based on the data which was generated elsewhere. Okay, so we worked out that that child there had basically a deletion, not of one nucleotide, but 184,000 nucleotides on a chromosome, which accompanied a gene which had previously been shown to cause that disease, diamond blackfan anemia. So we're sure that that's what caused the disease in that child. That was a 7-year diagnostic odyssey for the child with massive expense. You may have seen this in the Sunday paper recently. This is again one of the first patients we actually sent to the same company where we got the data back to try and work out what was wrong with that child. With the 2 previous pregnancies that this couple had had which didn't go to term. So there was nothing known that was wrong with these children, which had ended up being terminated. And we had to go well into this area and work out what that needle in the haystack was. And in fact, we generated an animal model of the disease which had killed the 2 babies of this family. So we actually became quite expert in analysing this data and in fact generating this data. And there was an alliance forming in Australia, and we've tried to form an alliance in South Australia as well. And in fact, we were the first in South Australia, SA Pathology, to receive accreditation, which is what you need to do these tests clinically, for what's called whole exome sequencing, whereas we look at those 20,000 genes at a time, which is quite something because lots of people in Australia wanted to be first to do that. And we're part of collaborations, not just in South Australia, but nationally and internationally, where we're trying to work out what's wrong with children, because we have 20,000 human genes. There's so far been roughly 5,000 human diseases caused by mutations in genes described, but there's 20,000 genes. There might be a lot more than that. Okay, so I've said that germline mutations matter from conception to old age. Let's have a bit of a look at what's happening in conception. So I've told you that as you age, you accumulate somatic mutation, and this ends up causing cancer. When you're born, it's frequently your germline mutations which might be responsible for whatever medical problems you have. And in fact, a huge proportion of admissions to Children's Hospital are caused by rare genetic disease. And so these obviously have, in the environment which that child is in, basically today's world, a very negative selective pressure. And in fact, a huge proportion of paediatric deaths under 5 years of age are due to monogenic disease. Okay, so as you get older, your somatic mutations accumulate, and again, you can either have somatic mutation, germline mutations, sorry, which might protect you from cancer, or they might predispose you to cancer, and we'll come on to that in a bit. So I'm going to have a quick look what happens here. Okay, in utero, from 13 weeks of age to 28 days after birth. So this is called perinatal death. And the underlying cause of death of fetuses in South Australia or in worldwide is actually unknown in roughly 50% of cases, which means you don't know what the chance of that happening for children, for couples is again. It makes assisted reproductive technology is not possible if you don't know what's happening. And there's guilt, self-blame, anxiety and depression in these families who are unable to have children or have repeated pregnancy loss. Now, I have no idea how people do some of these health economic analyses. PricewaterhouseCoopers seem to pluck numbers out of somewhere. Andrew, I didn't say where. And this was an estimation that they made a couple of years ago about the economic loss that stillbirth causes in Australia, which is quite incredible. So we've actually been sequencing stillbirths. So we're performing either whole exome sequencing or whole genome sequencing on undiagnosed cases of perinatal death to try and identify genes or the mutations which might be responsible for this if they are. But then we have to take it beyond our standard knowledge, and we need to actually prove causality of selected candidates using bioinformatics and both laboratory work as well. So if we find something, we'll have an answer for the families. We'll be able to tell them how to avoid having additional stillbirths or children who might be affected by a severe genetic disease, and they can use assisted reproductive technologies. The identification of new disease genes actually is quite stunning in terms of what it does in terms of biology as well, but that's taking it a little bit out of the personal. But working out what a gene actually does, so we understand some of the function of human biology, is pretty important as well. So this is 40 families that we've sequenced, and we sequenced the mum, the dad, and the stillborn child. And what you can see is that in roughly 30% of cases, we've solved what's wrong with those children, and so why this family is having stillbirths or unborn children. In another 30%, we think we know why. We're not quite sure. In another smaller proportion, we have multiple candidates. So we think it might be this one or it might be that, but we're not quite sure. So we need to go in and do lots of functional analyses to work out why. There's an awful lot of new knowledge in here, as well as helping the families. Roughly 30% of the genes that we're pulling out here were unknown to be caused in the phenotypes that we're observing in the fetus. So what does this mean for developmental biology? Basic developmental biology is absolutely massive. And there's a huge proportion of these as well where we actually have to go in and do functional studies. So it's going above and beyond our basic level of knowledge, which would be possible in a diagnostic lab. So already we've had 4 families using pre-implantation genetic diagnoses and 3 healthy babies. I think it's just 3. Has it gone up here again, Alicia? No? Okay, so that we know of. And this is the family which was on the front of the Sunday Mail a few weeks ago. And many of these families are actually reaching what you might call for the woman the end of their biological period. So it's pretty important to try and solve these as soon as possible so they can have a family. We're actually continuing to work on cases which we haven't solved, and we're actually about to take this whole study national, and we'll be sequencing 250 cases per year for the next 2 years at least. That's what we know we're going to be able to do. And our success rate of solving these is close to 60% at the moment, which is absolutely massive. We're still working on those unsolved 40%, and as I said, at least 30% of that is new knowledge, so that's amazing. So if you actually take children who survive that perinatal birth, the perinatal period, and actually present with a childhood syndrome at a hospital, how should you diagnose them now? Should you do this gene test by gene test? Should you run lots of repetitive pathology tests? Or should you do one of these whole exome or whole genome sequencings? So this is a study which came out of the Murdoch Children's Institute or the Royal Children's Hospital in Melbourne, where they took 101 children who presented in the clinic at birth. So you'll see that if they put them through all of their standard routine analyses, they ended up with 11% rate of diagnoses. When they use these new genomic technologies, they're getting roughly the same rate of diagnoses we're getting, 60%. You look at how much it costs to reach those diagnoses, $27,000 without using this genetic technology, and only $6,000 if they did use these genetic technologies. So 5 times more patients diagnosed, cost per patient's reduced 75%, and it actually changes what you do with those patients. It changes the treatment. Now, the change for the treatment isn't always fabulous. It may be that the treatment will stop with the child and they'll go into care until they die, but because we know there's nothing they can do for them. But, you know, to have a diagnosis and to be able to prevent this occurring again for some of these families is incredibly important. So it's faster, it's cheaper, it's better. Okay. What about later in life? Do we have any information later in life? What happens with people after they're presenting a bit later than at birth? If you're presenting with something from the perinatal period or straight after birth, you obviously have something pretty catastrophic which is going wrong. So maybe these ones are going to be a bit harder to look at. So for about the last 13 or 14 years, I've been collecting familial leukemias and familial hematological malignancies. Familial cancer is tragic, as I'll show you in a minute, and a quick summary of my career in this area would be, first of all, people said it didn't exist, then they said it was too rare to matter, then they said we have no idea what you can do with it, and then they said it has no relevance to anything else. So a quick summary of the rebuttals is it's a lot more common than they thought. It has a lot of implications for standard care of standard cancer patients. So, and in fact, studies that we have done have actually made classification of patients who are predisposed with leukemia into the World Health Organization classifications of leukemia. And why did people say that it didn't exist? When way back in 1861, if we've got any German speakers in the audience, you can see there were already publications saying that you did have leukemia running in families. And the first massive family or couple of families, actually 3 families, with familial leukemia were published back in 1999. So we started collecting families which showed these predispositions to leukemia, and we've got over 140 families involved. We've led to the identification of a couple of new genes. This is a list of all of the genes for these disorders which they said didn't exist. These are the types of malignancies which are running in the families. So myelodysplastic syndrome and acute myeloid leukemia. We have a lot of lymphoid diseases as well. So this is non-Hodgkin's lymphoma and Hodgkin's lymphoma. So roughly 25% of these families are solved. So what? So this is an Adelaide family. 13 or 14 cases of acute myeloid leukemia. Teach you how to read this. This is a woman, this is a man, this means they're married or they've partnered to have children. These are the children, a son and a daughter. Okay, so this guy here got acute myeloid leukemia, and the line through him means that he died. So acute myeloid leukemia is not normally that bad. It's not nice, But if you look through this pedigree, there's only one person who's affected with acute myeloid leukemia who's actually survived. And we actually worked out what the mutation in this family was, so a world first, and it was a mutation in the gene called GATA2. And if it says T354M, that means that this patient carries the mutation which predisposes them to leukemia. So they can get the leukemia young. Some of them are obviously still managing to survive and breed, but many of them aren't. They may have some other phenotypes which are associated with this acute myeloid leukemia. But one of the most tragic things with these families is that when an individual is identified, the first thing you do is you look for bone marrow transplant donors, and you normally look amongst their relatives. And so in this family, for example, this core family over here, a whole group of them were diagnosed roughly at the same time, simply because one of them got leukemia and then the rest of them were tested to see if they'd be suitable as bone marrow transplant donors, and they were all found to have leukemia. So the only survivor in here was transplanted aggressively at the first signs of leukemia. But this is the curious thing about genetics. It's not 100%. Your fate is not absolutely sealed by your genes. We've got two individuals who carry these mutations who should be predisposing them to leukemia, and they're well into their 60s, and they're well. Now, there's actually a lot of effort going in to working out why people in families like these who carry mutations which should make them sick are well. Why aren't they sick? Because there might actually be therapeutic benefit for lots of different people in working out why they are. And we've got a few clues on that, as you can ask me about later if you want. But what does this do to your headspace? This group here, they know they're at risk, and this is without having any genetic testing. They've all had their marrow frozen prophylactically to try and buy them a bit more time. Should they become sick, they can have that marrow put back into them to hopefully help them survive for a bit longer. That's actually this guy here, and this is his mum, and you say that's a pretty awful nuclear family outcome. So we actually worked out what was wrong with this family and introduced genetic testing for these before anyone else in the world. So this is another Adelaide family, similar type of history, except this time they don't just have acute myeloid leukemia running through the family, they have thrombocytopenia as well. And we noticed that they had skin disorders and arthritis running through the family. And then we looked at other families who had mutations in RUNX1, this gene RUNX1, and we found a number of families who, they didn't just have myeloid leukemia, they had lymphoid leukemia, but they also had psoriasis or eczema or skin disorders. And this individual here, down below, was actually treated with a very novel drug for psoriasis, and it totally cleared up her psoriasis. And in fact, we're now testing that drug to see if it works on acute myeloid leukemia, not just in families which have mutations in RUNX1, but any cancer which have mutations in RUNX1, which is actually up to 30% of all acute myeloid leukemias. So that's work actually in progress, where the clues actually came from studying a rare family, in fact a rare Adelaide family. So yeah, that's work in progress, which may lead to new drugs or repurposing of drugs. So if you actually have a germline mutation which predisposes you to developing a tumour, the tumour doesn't develop in isolation. So these are basically individuals from these families. Each column is an individual, and each line is a different gene, and every time you see red, it means that the acute myeloid leukemia which developed in those individuals actually developed a mutation, and that's why the tumour started progressing. If you remember, I said that cancer is often an accumulation of mutations. So what? Well, what do you do in a family like this? How do you tell what's going to happen to them? Well, the obvious answer is you try and work out whether you can tell when they're going to develop leukemia, and then see if you can intervene. And so this is basically showing this patient here over a number of years, and we can find a mutation which starts off at very low level in the blood and then progresses, goes to higher level as their disease progresses. So this is basically monitoring for pre-malignancy or molecular monitoring. Now you might have seen in the press circulating tumour DNA monitoring. Well, they've got an acute myeloid leukemia, so it is a circulating tumour, and it is DNA. So it's basically the same theory, is that can we detect when the disease starts to progress? And so this is one of the major questions. What would you do with these families is how you conduct surveillance on them and when you intervene. And by looking at these families more systematically, we're hoping we can answer those questions. So this is one of the latest genes that we worked out was predisposed to acute myeloid leukemia and myelodysplastic syndrome. But there's a major difference here between those other families. If you look at the age of onset of these individuals, they're old, right? So until the average life expectancy of our populations went up to this stage, you wouldn't have even seen that this mutation even mattered, right? Does it matter now? Well, yes, these people are getting leukemia. We think we know how to treat it better because they have that mutation. But it's making choices in people's lives. So, for example, one of the ladies here was used as a marrow transplant donor when one of her siblings got ill. And one of the ladies, I think it was this one here, her ovaries were used for an ovarian transplant with one of her daughters here. All right, so if you like, that's passing on the disease in a way which is probably less than desirable. Now, let's talk about myelodysplastic syndrome and acute myeloid leukemia. What we noticed when we looked at other tumours which occur in this cohort is that we do have other tumours. And in fact, if you look at non-Hodgkin's lymphoma and Hodgkin's lymphoma, they tend to cluster together with tumours like breast cancer, prostate cancer, and melanoma. And when we start sequencing these families, we start to find mutations in genes such as PALB2, which is a known breast cancer predisposition gene, okay? It's not BRCA. We do have families with BRCA mutations in them as well, which is the Angelina Jolie gene. But this is another breast cancer predisposing gene. So here you can see our family who was ascertained with Hodgkin's lymphoma over here, okay? And when you expand the family tree out, you can see on another side of the family they've all got breast cancer. And a similar case down here where we've got diverse types of cancers occurring. So what does that mean? That actually means that this tumour streaming which people have been performing, saying you've got breast cancer and you're predisposed to breast cancer, or you've got colorectal cancer and you're predisposed to colorectal cancer, while true is not completely true, which means that you might want to be able to monitor not just for breast cancer, but for Hodgkin's lymphoma in these families. And how do you do that? And this can, again, alter the treatment of these families. So how can it actually alter the treatment in these families? This is actually what's called the DNA damage repair pathway. And there's new drugs called PARP inhibitors, which are working in these DNA repair pathways for treatment of patients as we speak in both clinical trials and routine care. So the large hope of genomics is that it will have massive implications for health, but particularly cancer. So at the moment, there's a massive group of cancers that we just lump them together and we give them radiotherapy and or chemotherapy, and they fall into these different groups. And some of those groups are desirable and some of those groups are not desirable. So drug not toxic and beneficial, you'd love to go in that group because that's curing you. Drug toxic but not beneficial, that's probably your least favourite group. Okay. Now some of the newer drugs which we're getting out for cancer are a lot less toxic than some of those common treatments, but how do we know who should be treated with which drug? And in fact, when you look at cancers, every year in Australia there's 52,000 diagnoses of less common cancers, and they actually cause 25,000 deaths. So these are all the big cancers: acute myeloid leukemia, breast cancer, prostate, bowel. And these things are called less common cancers and rare cancers. So here's research dollars being spent, total cancer burden of disease, and the number of deaths. And you see across in this rare corner, there's very little money being spent on less common and rare cancers, which actually all of my families fall into as less common and rare cancers. The burden of cancer death and the cancer burden is actually very high. Okay, And there's probably over 180 plus cancers as defined by various traditional pathology techniques within these groups. So the solution for these types of things, we think, is molecular screening and therapeutics. And so this is a clinical trial which is happening, it's called MOST for M-O-S-T, and a number of clinical studies which are going on. So if you take these rare tumours and you sequence them with a limited panel, you can find what we call actionable mutations in greater than 20% of these patients. So instead of offering them just standard chemotherapy and radiation therapy, they can go on to very specific therapies. If you don't find an actionable mutation, you can put them onto an immunotherapy, which people might have heard about, which is a much more recent and very effective therapy. Here's that group that we talked about, PARP inhibitors for DNA repair, because this is becoming a lot more common than we thought. So there was a report issued in January this year by Greg Hunt, the Federal Minister for Health, in association with Rare Cancers Australia, and the recommendations were that we should be able to have local clinical trials to test and confirm local clinical trial design. We should have subsidised access, and we should have collaboration to infrastructure. So they want to have a network where patients with cancers can be fed into the appropriate clinical trials. This actually became government policy for one party. That party didn't do very well, I'm afraid. So if any money follows through, who knows? But if you actually look at cancer more broadly, and this is data from David Thomas at the Garvan Institute, if you take over 300 cancers at presentation and you sequence them, you can find actionable variants in at least 46% of them. So what that means is that you can find a new or a different way of treating them compared to standard treatment in almost 50% of them. And if you can access the appropriate clinical trial, here MOST, for example, you can get a significant proportion of those patients onto a new, less toxic therapy, which will hopefully, than chemo or radio, which will hopefully, if not cure them, at least make them survive with a greater quality of life, hopefully for a longer period of time. So this is the progress of that molecular screening study to date. So here's South Australia, okay? We don't have funding to do this. Here's the Kinghorn Cancer Centre and the Chris O'Brien Lifehouse in New South Wales, who are the lead contributors to this study. So if you ended up with a rare cancer, I'd ask you the question, where would you want to be? Would you want to be in Sydney, or you'd want to be here? And I think the answer is pretty obvious, which is why you want your governments and your researchers to be invested in this type of problem. So we are part of a national network here with RA and the Centre of Cancer Biology and Assay Pathology, trying to get this up and going for South Australian patients as well. But we're funding dependent. We're hoping that money's going to come from the feds, but we also need contributions from the state government. So, which takes us to our South Australian Genomic Health Alliance. So here's the investment that various countries and states have made in genomic medicine, if you like, the investment of sequencing people, sequencing tumours. And what you can see down the east coast is that each state government has put in relatively large amounts of money, not nearly as much as England or America, but relatively large amounts of money into trying to make genomic medicine part of their standard healthcare. That red dot here in South Australia is a hopeful question mark, and there has been substantive federal government funding, of which we do get some of that in South Australia, but it's not nearly enough. So our Genomic Alliance in South Australia is basically to help develop better value healthcare through genomics. We want to be visible. We want to make sure that genomic services in South Australia are state of the art. We want to make sure that we're keeping research activities and innovation right up to the cutting edge to make better healthcare for patients. So this was helped a few years ago by a large infrastructure grant from the Australian Cancer Research Foundation, where we opened a genome facility. We had additional money from the state government and some other bodies, including University of Adelaide. And this has set up an ecosystem, but this ecosystem still needs more support. where we have basic research and translational research where we're diagnosing patients, whether they be with genetic disease, predisposition to cancer or cancer. And we can end up with a clinical diagnosis that can end back going to a clinical trial or can end up going back to basic research because of discovery of a basic biological mechanism. So this is the ecosystem that we've tried to set up and support for the benefit of South Australians. It has a flow-on effect. So since that genome facility was opened, these are the number of grants that that genome facility with our expertise has actually been consulted on to try and attract additional grant funding from the feds or from other organisational bodies. And you can see it's been pretty significant. Bringing that expertise into South Australia has brought extra research dollars into South Australia and extra expertise into South Australia. So we believe instead of saying that someone else is going to do it, it can all be done in Melbourne, it can all be done in Sydney, it can all be done in China, it can be done cheaper and better. These are the types of benefits that you get from doing it here. And actually, if you look at the number of samples which have been run through that genome facility, these are the number of research samples which have been run, and these are the number of diagnostic samples which have been run. And you see that we're clearly converting the latest technologies from being research only to being diagnostic as well. So, for me, this all seems bleedingly obvious. We should be sequencing everyone when they're born, see what's wrong with them, what they're going to get. If you get a cancer, we should be sequencing it and see how we can treat you. So why aren't we? There's a lot of ethical concerns because what you have in your germline also affects other people in your family, amongst other things. Do you have the right to know what the future of your child is going to be based on what they have in their genetics? The economics is horribly frustrating for me. Who pays for the testing? A very simple question. Now, for some of these rare genetic diseases, if you actually come back as having a positive test, there are treatments available. Not for very many of them, but if there is a treatment available, that treatment might cost hundreds of thousands of dollars a year. So who's going to actually pay for them? And then you have this catch cry throughout medicine called evidence-based medicine. And what they want to know is, does it alter clinical treatment? Does it save lives? Does it save money? But by the time you actually accumulate all that evidence, it may be too late for you, your loved ones, and your family. And then we have the question of education, is that people don't know it's available, they don't know how they can benefit, it can benefit from, and the medical profession is not totally aware of all of these things as well. So I leave you with that final thought again. My main message is that embedding research with standard healthcare is vital. If you want to have access to the best standards of healthcare possible, then tell your politicians that you think we need extra funding. Get ready for your grandkids to be playing in their bedroom with a thumb prick. They're not doing their diabetes anymore. They're actually sequencing their genomes and posting it on Tinder. And I'll leave it there and be very happy to take any questions. Thank you.
Thanks, Hamish. Fabulous lecture. This is open for questions. Maybe I'll start by, you mentioned early on about some of the limitations around interpretation of the data, and that talks to the kind of bottleneck about bioinformatics. With artificial intelligence, machine learning, are we quickly going to that situation that will be on the iPhone and so on?
I wouldn't say quickly, but yes, eventually. I'm actually trying to prepare a document at the moment which is outlining to medicos who might be referring in Australia, South Australia, about any genetic testing, why this should remain in the public system, why this data should remain in the public system, because if it goes into the private, goes into a database, they might issue a single clinical report, but all the rest of that data is lost. So how do we get to a state where you might have artificial intelligence helping us? You have to have very large-scale databases, which are going to require cloud infrastructure and detailed data, which won't come solely from private. It needs public investment in it. So you can imagine that you should have your genome sequenced at birth, and this should become of your one of your health records. I actually do have my genome on my iPad if anyone wants to look at it. Actually, I don't think I've got my iPad with me. You can look it up on Tinder though, and it's perfect. So if you want to reproduce, please see me.
I think we'll pass on quickly there. Yes, please.
I was curious about some of these monogenic conditions that you're talking about, where you're treating with drugs. What do you think about some of these technologies that they're using with CRISPR and the like, in terms of actually, does that allow these familial conditions to actually be wiped out of the family, or is that then having to look at gamete changes and so forth?
Well, that's the million-dollar ethical question. So I know ethicists who would like to be controversial and saying, why wouldn't you? Why wouldn't you wipe out one of these diseases if you could? I can tell you that CRISPR has already entered the clinic, and the child I put up there with diamond blackfan anemia, he could probably be treated with CRISPR technology at the moment, but there'd be risks associated with it. But that you would not go in and touch his germline, so you wouldn't affect his future at the moment, his future offspring, and he might have children. So that's a massive ethical question which has yet to be answered. You know that in some genetic communities there's it's very interesting. So the deaf community, for example, there's resistance to have cochlear implants because they think they're a special culture, and that, well, they are, and they don't want to lose that culture. There's non-invasive prenatal diagnosis available for a lot of genetic diseases at the moment, but it's come to the fore for Down syndrome. And so, if you like, the elimination of Down syndrome has actually become a public health goal of lots of countries, and it's doing pretty well in some countries. I don't know, it was National Down Syndrome Day the other day. Any of you see that car karaoke with the Down syndrome kids? Look it up. Should we be eliminating these things, or should we just be living with them? I'm not sure. I think it's a very personal question. So I prefer to leave it to people rather than make a... But touching the germline, I'd be very, very cautious.
Yes, please.
We've seen these certain tubes sort of test 23andMe and Ancestry DNA and Prometheus. What can you say about the accuracy of those, or the darts on the dartboard sort of thing?
Many of those are doing what I call recreational genomics. And that's fun. There's a whole bunch of websites out there that they generate different types of genetic data, okay? So it might not be as complete as having a whole genome. So it might only access a certain number of bits of the genome. But there are websites out there which you can upload them and see who you're related to. So if you're an Ashkenazi Jew, you'll be related to Sergey Brin. You might discover long-lost cousins. You can assay, you can work out how athletic you're meant to be and probably be disappointed because you're not. Or, so there are these recreational genomic sites out there. I think it's fun. If you want to have the money and you want to spend it on that, go for it. Ancestry.com is an absolute classic, and you see the ads on telly. I think it's fun. I think it's a great educational tool. 23andMe got into a lot of problems when they tried to sell it and provide medical information, basically because the FDA in America wasn't convinced by the veracity of their medical advice because they didn't have decent oversight of it. And I think that's the problem, because most genetic tests, particularly when it comes to germline, are given with significant amount of genetic counselling explaining to you exactly what this means for you and your family. And if you don't have that counselling, Dr. Google's not bad, but it's not perfect either. So it's dangerous. Education is a massive part of this. So I quite like recreational genomics, but I'm frightened of it at the same time. You know, I walked into my GP's office after I had my genome sequenced and said, you know, I had my genome sequenced, and his interest level was a big fat. Hopefully that'll change, because there are some things in my genome which maybe they're not quite perfect, shocking as it may seem.
You've just lost that previous offer, haven't you?
We can CRISPR correct them, it's okay.
Andrew.
Just a question around actionable target development. So the idea that you can identify targets that are currently non-actionable by drugs and so forth. Do you see this as a big drive of pharma development of new compounds that target recurrent mutations or recurrent genes that are mutated in individuals with particular disorders?
I'm going to ask you to restate that question in a different way. I didn't quite understand. In English would be good.
Okay. Actually, this is going to be interesting. with sort of, I guess you're identifying new genes that are related to particular disorders clearly continuously in a research setting. Do you see that as being one of the new drivers for drug development by pharma in sort of identifying current target or current genes that are non-actionable because they don't have compounds to treat with? Do you see that as a big driver in pharma?
It's definitely not at the moment. So will it be? I don't think so, not until the biology behind some of those genes is a bit more sorted out. Yeah, the funny thing is that amongst some of the known genes which are popping up in that perinatal death cohort, for example, there's some old favourites like iduronidase, mucopolysaccharidosis type 1, but there's also probably a significant number of cancer predisposition genes, so where the fetuses are being being miscarried. So driver of targets, I think there's some systematic efforts out there. Will this help? Maybe, but the problem, so one of the major problems with personalised medicine, whether it be for a germline or a somatic, is often you're talking about n equals 1, right? And I mean, I didn't present, but there's one case here where we found a defect in a vitamin transporter, and we gave the child vitamins, and it was massive in clinical improvement. It's n equals 1 though. And many of these cases are n equals 1. Until you can prove the relevance of the biological pathway to something significantly broader, I don't think there's much interest from pharma. So even that one case where we've probably shown that a Genentech drug for psoriasis is relevant to acute myeloid leukemia, we're interested. People with RUNX1 mutations are interested. Genentech's not that interested, which I don't really understand. But
okay, I think we'll need to draw it to a close. So, Hamish, thank you very much for demystifying genomics for a general audience. I think you've given us a great oversight of the opportunities, but also some of the challenges of modern-day sequencing. There is just a couple of minutes left if people want to queue up who have a lazy 1.4 million for your new sequencer. But thank you, and thank you all for coming. I've just have one parting token of appreciation, one of which may have some epigenetic effects, but thank you once again. Yeah, cheers. And we look forward to seeing it for future Health Matters lectures. Thank you.